Ladies and gentlemen, thank you very much for attending this scientific evening. We're very pleased tonight to have Professor David Liu, the winner of King Faisal Prize in Medicine 2022 with us, to give 2 lectures. And let me just start by congratulating David for winning the well-deserved prize. And I will just give a very short introduction of David. He is the Richard Merkin Professor Director of Merkin Institute of Transformative Technology in Healthcare. He's also Vice Chair of faculty at the Broad Institute of Harvard and MIT, and Professor of Chemistry and Chemical Biology at Harvard University.
Hello?
And Harvard Medical Institute.
Yeah, it looks good. Yes,
David,
good. Thank you.
Graduated first in class at Harvard in 1994, and then earned his PhD at UC Berkeley in 1999, then moved back to Harvard University. He's an academic science advisor to the US government. He published more than 165 papers and is the inventor of more than 60 issued US patents. In 2016, he was named as one of the top 20 translational researchers in the world by Nature Biotechnology. And in 2019, Prime Editing was named one of Nature's 10 remarkable papers from 2019 and one of scientists' top technical advances. He has numerous teaching and research prizes. Last but definitely not least, the King Faisal Prize. So please, Professor Liu.
All right. I'm really grateful for this opportunity to share some of our work with you. I know I speak for both of my guests as well as certainly for myself that this experience of the King Faisal Foundation, the ceremony, and maybe most importantly getting to know this remarkable culture, being treated so warmly and so well by everybody has really been very meaningful to us and has made this trip very special. So thank you so much for that. So I'll give 2 lectures. The first lecture will be about the development of 2 of the gene editing technologies that are recognized in the King Faisal Prize citation, which are base editing and prime editing. And then in the second lecture, I will describe how these technologies are being used to treat animal models of human genetic disease. And I'll give a little update on efforts to advance these technologies towards the use in human patients as well. So much of our lab's work is motivated by an examination of the different ways that mistakes in our DNA can cause genetic disease. This is a fairly representative pie chart of all the different ways that mutations can cause genetic diseases. And you can see that there are 4 transition mutations, which are these 4 single-letter DNA swaps that result in diseases like progeria, the rapid aging disease. There are 8 transversion mutations in which a purine and a pyrimidine switch positions. These result in diseases like sickle cell disease. And there are also, of course, deletions, like a 3-base pair deletion that is the most common cause of cystic fibrosis. There are insertions, like the 4-base insertion that causes Tay-Sachs disease, and many, many other variants. So it's, of course, been a longstanding goal of the life sciences to be able to install or correct as many of these different kinds of mutations as possible so that we can study or potentially treat the broadest possible range of genetic diseases. Now, the era of modern gene editing really began with programmable nucleases. These are DNA cutting enzymes like zinc finger nucleases, tail nucleases, and of course CRISPR-Cas9 that was recognized by the 2020 Nobel Prize in Chemistry. These programmable nucleases cut the DNA double helix into two pieces, and they really initiated the modern era of gene editing, but it turns out that they are not well suited to install or correct the vast majority of mutations that cause genetic disease. So why is that? Well, the reason is that when you cut the DNA double helix into two pieces using a nuclease like CRISPR-Cas9 or a zinc finger nuclease or a tail nuclease, in mammalian cells, the primary response to that cutting is the initiation of end-joining processes that are the cell's attempt to get the ends of the cut DNA back together. And usually the ends come together perfectly, but then the nuclease, the scissors, can cut the DNA again because the sequence hasn't changed. And so this process occurs over and over again until the cell makes a mistake that causes the sequence of the DNA to change so that it's no longer cut by the scissors. So the result of this process is that programmable nucleases result in mixtures of insertions and deletions that we can't control. We can predict them, they're reproducible, but we can't control them. And so the net result is that nucleases, the DNA cutting enzymes, are primarily useful for disrupting genes because making insertions or deletions most of the time will render a gene inactive or a gene product inactive. In addition, when you cut a chromosome into two pieces, you also cause other effects in the cell, most of which are undesired, especially for therapeutic purposes. You cause sometimes the loss of a large segment of the DNA. You cause chromosomal translocations. You activate DNA damage responses that can lead to the enrichment of oncogenic cells. And so these are also downsides of making these cuts in DNA. All right, now seminal work by Maria Jason and other scientists showed that you can use a nuclease to stimulate not just targeted gene disruption, but also precise gene correction through a mechanism called homology-directed repair, or HDR. This process uses a donor DNA template to replace the DNA sequence around the cut site. Unfortunately, we now know that HDR is inefficient in most types of cells, including almost every kind of cell that is therapeutically relevant. And so, as you might expect based on the slide, if you look at the CRISPR clinical trials that are underway, almost all of them are trying to benefit a patient by disrupting a gene. Very, very few of them are trying to precisely correct a gene using a CRISPR-Cas9 nuclease because this process is so difficult, so inefficient in most types of cells. Okay, so really the subject of these talks is the result of our efforts to develop ways of precisely correcting genes, not just disrupting genes, without cutting the DNA double helix, without cutting a chromosome into two pieces. And some of the key developments in this quest were the development of the cytosine base editor, which makes these two kinds of specific single-letter changes, the adenine base editor, which makes these two kinds of changes, and the prime editor, which makes a wide variety of change. Almost any kind of change that you can make in a small segment of DNA can be installed by a prime editor. Now, they all have their strengths and weaknesses, some of which I'll be able to talk about. But that's sort of the overall summary of the main technologies I'll talk about today. And even though we developed base editing in 2016 and prime editing in 2019, as of today, these remain the only general, programmable, reasonably efficient ways to directly correct mutations in most kinds of human cells. Okay, so the cytosine base editor was the first base editor. This was a engineered molecular machine that was developed by a former postdoc in the lab, Alexis Comore, who's now a professor at UCSD. In the interest of time, I'll just summarize by saying that a cytosine base editor consists of three components, three proteins. There's a disabled CRISPR-Cas9 protein that can no longer cut the DNA double helix, but it retains its ability to bind to the guide RNA and to a target DNA sequence in a guide RNA programmed manner. The protein in red is a cytidine deaminase enzyme that we took from nature, which catalyzes the chemical conversion of C into U. And U, of course, is a base that pairs and is copied like T. And then the third protein is this uracil glycosylase inhibitor, which plays a really important role in guiding the cell through the DNA repair processes that result in making this conversion permanent on both DNA strands. So the way a cytosine base editor works is the base editor binds to the target DNA sequence in a guide RNA programmed manner. And then within a small window of typically 4, 5, 6 base pairs, cytosines are turned into uracils through the action of the red protein. Now that creates this GU mismatch, and the GU mismatch will be resolved by the cell. Now, most of the time the cell will, your cells have an enzyme called uracil DNA glycosylase that will very quickly cut out the U and result in the reversion of this edit back to the original CG base pair. But in the second and third base editors that we developed, BE2 and BE3, we use this purple protein to slow down this process. And then in the BE3 system, the base editor nicks the non-edited strand containing a G. And that nick stimulates in eukaryotic cells, stimulates cells to remake this DNA strand, replacing the G with an A. Because if you remake this piece of DNA when a uracil is at this position, then of course when the polymerase fills in the remade strand, it will install an A. And once you're at a UA, then really the only thing that the cell can do is turn it into a TA base pair. So whether it's UA or TA, the job is basically done. And you've converted now a CG base pair to a TA base pair. And so if you just take dead Cas9 and fuse it to a cytidine deaminase, you get a sort of very poor base editor shown in red. If you add the uracil glycosylase inhibitor, the purple protein, you get a better base editor. And if on top of that you nick the non-edited strand, you get a much better base editor. And all of these were more efficient, at least at the time, than simply using Cas9 nuclease and an HDR template to try to install this single base pair swap. So the cytosine base editor can make this kind of change, which turns out to have the potential to correct about 14% of known pathogenic point mutations. And the point mutations represent about half of all known disease-causing mutations, period. But you'll notice that one particular point mutation is disproportionately represented among all pathogenic human point mutations, and that's the mutation that can be corrected by converting an AT base pair to a GC base pair. So we set out to develop an adenine base editor to accomplish that chemistry. And if you deaminate adenosine, you get inosine, which is read as a G. So that is a potential chemical strategy to achieve this goal. And so we imagined that maybe you could take a deoxyadenosine deaminase enzyme, fuse it to a Cas9 nickase, and basically do what we did to develop the cytosine base editor. But now you would convert an AT base pair through this process to a GC base pair. But the biggest problem with this idea is that nature does not provide any enzymes that perform this chemistry on DNA. Nature provides adenine deaminases that work on RNA or on other kinds of adenine bases. but not on DNA. And so we decided, led by a former postdoc in the lab, Nicole Godelli, who's now at Beam Therapeutics, that we would try to evolve our own deoxyadenosine deaminase enzyme. And we really didn't know if this would work. But Nicole set up a Darwinian selection that allowed hundreds of millions of different variants of an RNA deaminase enzyme to be mutated and to be selected for their ability to deaminate adenine in DNA. And their reward for doing so was survival, the ultimate reward, survival in the presence of an antibiotic. And then she transplanted these deoxyadenosine deaminases that she had just evolved into a mammalian cell base editor architecture and hoped that we would achieve the first conversion of an AT base pair to a GC base pair. And the project was successful. When you start out by just fusing RNA deaminase to a Cas9 nickase, you get no editing. But through 7 generations of evolution, she evolved these mutations that collectively allow this enzyme to deaminate adenine in DNA, to deaminate deoxyadenosine. And that turned into the first adenine base editor. Now, base editors have since been improved and studied in many, many ways. One of the most important ways that they've been studied and improved is to understand off-target base editing and to engineer new base editors that minimize off-target editing. And that off-target editing can arise from several different sources. The Cas9 protein, or the CRISPR-Cas protein that we use to target the DNA, is not perfect. It will bind sometimes to sequences that are very similar to, but not identical to, the target sequence. And researchers have developed a number of high-fidelity Cas9 variants that can be used to create high-fidelity base editors that minimize this kind of off-target base editing. A second kind of off-target base editing can arise from the deaminase enzyme independently operating on DNA. This is called Cas-independent off-target DNA editing. And the initial cytosine base editor had enough of this activity that even though it was fairly rare, you could detect it by whole genome sequencing with a frequency that is about 5 times 10 to the minus 8th per base pair in mouse embryos that received high doses of base editor. But subsequent generations of base editors have minimized this kind of Cas-independent off-target DNA editing, often down to background levels that are comparable to untreated cells. And then finally, you can also get off-target RNA editing, because these deaminases, in some cases, will still deaminate RNA. Now, you may not worry about RNA off-target editing because the RNA is transient after all. But we also, our lab and other labs have also worked to engineer or evolve variants of base editors, deaminases, that reject RNA and have much lower off-target RNA activity. Okay, so that's a summary, very briefly, of many, many studies from many labs, some of which are summarized here, that show how off-target base editing has been studied and minimized. Now, another way that we have advanced base editing is by evolving more active base editors. And to do this, we use our phage-assisted continuous evolution platform. This is basically a Darwinian evolution platform, but instead of taking a week or a month maybe to do one complete generation of making a library, selecting for desired variants, and then isolating the winners and then mutating them and repeating the cycle, PACE takes place very, very, very quickly because all of these steps take place during the life cycle of the filamentous bacteriophage, which can complete in about 10 minutes. So PACE is a very, very rapid way of continuously evolving proteins without human intervention. So we can set up the evolution, it can go very, very quickly, and then we come back the next day or maybe in a few days, and the genes that we're trying to evolve have had the opportunity to evolve the desired activity. So we set up a selection circuit that links base editing to the survival of phage that encode the desired base editors, which are represented in these red circles. And by setting up this genetic circuit, we could very, very rapidly evolve base editors. And the first thing we did was evolve them to be more active, to work faster. And this evolution was very successful. So out of about 36 generations of evolution, which normally would take you about a year, but takes place only 1 or 2 days, we ended up evolving 22 mutations total versus the original natural enzyme we started with that only works on RNA. But you can see, comparing the orange line to the black line, that the resulting base editor is now about 1,000 times faster. So this enzyme is so fast that the first data point that Jennifer Doudna's lab used to assay this new base editor, which used to be assayed at about 1 or 2 hours, all of the substrate was consumed. So the student in Jennifer's lab had to redo the kinetics, now using much, much finer time points, because this evolved base editor is 1,000 times faster. It's now so efficient that you can get 100% editing in cell types that are easily transfected, meaning ones that you can deliver the base editor into well. And in collaboration with Jennifer Doudna's lab, we solved the cryo-EM structure led by Jennifer's group. So this is the first 3-dimensional structure of the base editor, is this evolved ABE8E. And it shows in beautiful molecular detail how the mutations allow DNA to be deaminated and why they work so much faster than the original enzyme. Another way that we have expanded the usefulness of base editing using protein evolution is by evolving the Cas protein. So that was the deaminase. Now we've evolved the Cas protein as well. And a major requirement for base editing is that a PAM sequence, which is the short DNA sequence that a Cas protein needs in order to land on DNA to begin with, we evolved Cas variants that use different PAM proteins, different PAM sites, not just the PAM sequences that natural Cas proteins prefer. So once again, we developed a PACE circuit that allowed us to specify which PAM sequences we wanted to evolve Cas9 variants to recognize. And out of this process came 3 families of Cas9 variants that now recognize PAM sequences that are not NGG, the canonical starting PAM sequence of Cas9, but instead collectively allow you to target almost any NR, where R is A or G. So this makes the base editors that use these Cas proteins much, much more flexible, so that before, if you limit yourself to NGG PAMs, you can only target about 25% of the genome. Using these new flavors of Cas9 proteins that can recognize different PAMs, you can, in principle, target about 95% of the pathogenic alleles, pathogenic point mutations that can be corrected using base editors in the genome. All right, so mitochondrial DNA is very important. Our cells, each of your cells have dozens or hundreds of mitochondria and dozens or hundreds of copies of mitochondrial DNA in every mitochondria. And the mitochondria have this separate genome of about 16,000 base pairs. And mutations in mitochondrial DNA cause many genetic diseases as well. So researchers have long sought to be able to edit mitochondrial DNA. But that's proven to be very difficult. You can selectively destroy mitochondrial DNA with a nuclease, but when you cut DNA in the mitochondria with a nuclease, that copy of the mitochondrial DNA that you just cut just disappears. It gets degraded. So you haven't really edited mitochondrial DNA. You've just degraded targeted mitochondrial DNA. And that can still be useful for some applications, but ideally, instead of targeted mitochondrial degradation, you really want to edit, to specifically convert one DNA sequence to another in mitochondria. Now, one of the main reasons why this has been so challenging is there is no CRISPR that can be used in mitochondria. Otherwise, we would just use a base editor like I just described. The problem is that researchers haven't figured out how to get guide RNAs into the mitochondria. It sounds like a trivial problem, but it has been unsolved despite decades of effort. So instead, researchers, as I mentioned, the best they can do is selectively degrade target sequences. So Joseph Moguse's lab at the University of Washington, his lab studies bacterial toxins, he emailed and called me one day and said, I've discovered something that I think you might find interesting. And what he discovered was this purple protein called DDDA. DDDA is a bacterial toxin that kills other bacteria. And when he solved the structure and looked at the sequence of this protein, he realized it was a DNA deaminase. But when he did the usual biochemical assays on this deaminase, he realized that it does not work on single-stranded DNA, unlike all previously discovered DNA deaminases. Instead, this DDDA deaminase works on double-stranded DNA. So he called me and said, I'm not sure what we can do with such a deaminase, but I suspect that you might have a good use for it because you've been using deaminases for base editors. And indeed, we told him we know exactly what we want to do with that. So the two base editors that I told you about all use CRISPR-Cas9 to target DNA. And because CRISPR-Cas9 has to open up the DNA double helix so that one DNA strand can pair with the guide RNA, that leaves the other strand of the target as a single-stranded DNA, and therefore it needs to be deaminated with an enzyme that works on single-stranded DNA. That's great because all natural deaminases and our laboratory-evolved deoxydeaminase work on single-stranded DNA. But the reason we are reliant on CRISPR for these original base editors is because these deaminases only work on single-stranded DNA. So once Joseph shared with me that he had discovered a double-stranded DNA deaminase, it became clear that the best opportunity to have an impact for this double-strand deaminase in the field of gene editing, at least, was to allow us to now use CRISPR-free all protein base editors by using programmable DNA-binding proteins like zinc fingers or tail proteins to target DNA without, therefore, relying on CRISPR or guide RNAs. And so that's exactly what we did. This was a three-way collaboration with Joseph Moguse's lab, our lab, and Vamsi Mutha's lab. Vamsi is actually a former winner of the King Faisal Prize in Medicine. And what we did was first make this very toxic protein non-toxic by cutting it into two pieces, a light purple half and a dark purple half. Then, led by a graduate student, Beverly Mock, she fused each of these halves onto a tail protein array. And for those of you who know about tail repeat arrays, they're very special proteins because they allow you to target virtually any DNA sequence by simply changing two amino acids in a repeating protein motif called a tail repeat. So you can program a tail repeat array that will bind to almost any DNA sequence you can choose. And what we imagined is that if we fused each of these halves of DDDA to two tail proteins that were programmed to bind to adjacent DNA half sites, that they might reassemble the complete DDDA deaminase right at that target site, and that would allow the deamination to take place. But before that, the toxin would be cut in half so it wouldn't be toxic to the cell. And this turns out to be the case. After a lot of hard work, Beverly optimized the architecture of a mitochondrial base editor into a form that looks like this. And she used it to create the first purposeful changes in the sequence of mitochondrial DNA in living cells. It will also edit nuclear DNA, but of course, base editors that edit nuclear DNA are highly optimized, so they're generally not as efficient. But these mitochondrial base editors allow you to uniquely edit the mitochondrial DNA, which we could not do before. So here's a variety of different edits that we installed at genes that were of biological or even biomedical interest in mitochondrial DNA in human cells. The off-target editing from these mitochondrial base editors is very low. You can now study the effects of mutations, either installing pathogenic mutations or correcting them in the genome of mitochondria in human cells. And as one example, we installed a particular mutation that is in one of the proteins involved in aerobic respiration in the mitochondria. And the result of this mutation is the decrease in various cellular respiration functions, as well as the abundance of certain proteins in the electron transport complex. And researchers have now used these mitochondrial base editors to produce animal models of mitochondrial human diseases for the first time by installing exactly the mutations that you want in mitochondrial DNA. So that's a brief tour through the development of some of the base editing technology. Base editors have become very widely used. I get weekly reports from AdGene, which is sort of the nonprofit library of DNA blueprints, and they tell me that more than 12,000 of our base editor constructs have been requested by more than 3,000 laboratories around the world. And according to PubMed, more than 2,000 publications using base editing have now been published. Now, the base editors that I talked about can make these 4 kinds of changes. And those 4 kinds of changes are the most common kind of pathogenic mutations, but collectively they still only account for about 30% of the entire pie chart that I showed at the beginning of my talk. So we became very interested in developing new kinds of editing technologies that could also directly correct rather than disrupt genes, but could make transversion mutations or deletions or insertions. And that's where Andrew Anzalone, who's in the audience here, former postdoc in the lab, he took on this challenge, and his postdoc involved the development of a new kind of mammalian cell gene editing technology that could make just about any kind of local DNA change at a target site in a human cell, again, without requiring double-stranded cuts of DNA, because we don't want to make lots of indels and we don't want to trigger the undesired consequences of cutting chromosomes. So what Andrew developed was prime editing. And the way prime editing works is summarized in this slide. So prime editors are fusions of a CRISPR-Nickase protein with an engineered reverse transcriptase. And that fusion will bind to a target DNA in a guide RNA program manner. But instead of using a normal guide RNA, Andrew engineered what we call a prime editing guide RNA, or a PEG RNA. These are engineered guide RNAs that not only encode the target DNA sequence, but they also encode, using a 3' extension at the end of the guide RNA, the desired edit that you wish to make. So how do they work? The Cas-Nickase reverse transcriptase fusion nicks target DNA strand, and then the free 3' end of that nicked DNA strand hybridizes to a primer binding site that's been carefully engineered into the PEG RNA. So that forms this primer template complex. And now the engineered reverse transcriptase can polymerize DNA one base at a time using the rest of this PEG RNA's extension, the reverse transcriptase template, to encode the edit. So you can now encode the edited bases in red here, and the reverse transcriptase will directly write those edited bases into the target DNA. The result is a sort of bizarre-looking DNA structure in which you basically have the original DNA double helix, except one of the strands now has a 3' flap coming off of it that has the edited DNA sequence. Fortunately, these flaps are very common intermediates in DNA replication and DNA repair, and the cell deals with these flaps by equilibrating them and selectively excising 5' flaps. The 5' flap is the strand that was the original strand that does not contain the edit. So if the cell cuts off the 5' flap selectively and ligates the gap, then you end up with heteroduplex DNA, where now you've edited one of the strands, and you have one unedited strand. And now we know how to fix this because we can do what we discovered with base editors. We can nick the non-edited strand, and that will stimulate the cell to replace the non-edited strand with the edited DNA sequence. And now you have a permanent prime-edited DNA. Now you have complete control over the sequence of this reverse transcriptase template. So you can make it a little bit shorter or a little bit longer than the original DNA sequence, and that's how you can make insertions or deletions. So indeed, Andrew found that prime editing was a very versatile way, really unprecedented in the kinds of mutations that we could now directly install into human cells without making double-stranded cuts, without relying on homology-directed repair. One of the early experiments he did was make all 24 possible single base pair changes in a stretch of 8 consecutive bases in the human genome. And you can see he made all of them. And generally the efficiencies were decent, and there was a good ratio of edit to cells. You can do this at target sites of your choosing. You can make precise deletions, and you can do combination edits. You can insert 2 bases and delete 2 other bases, or you can do a 2-base deletion and a 1-base transversion. These are all possible now because of the highly versatile mechanism of prime editors. Another remarkable feature of prime editors is that contrasts with homology-directed repair, the other way to make changes in DNA of your choosing in a very versatile way, is that prime editing products are very homogeneous. So this is the sequence of 2 sites in the human genome in 2 different cell lines with no treatment. And you can see that there's some heterozygosity in the HeLa cells, but the K562 cells are pretty much one sequence at this site. Now, if you treat them with a prime editor, you get the desired product, which is in green, and you get almost nothing else. What I'm showing you here is every sequencing read that was detected at a frequency of 0.2% or higher. If you repeat this experiment using a Cas9 nuclease and an HDR template, here are all the products that have a frequency of 0.2% or higher. In other words, cutting DNA with a nuclease will inherently generate a mixture of these products. They will include the desired product if you're using homology-directed repair and you include a template, but most of the products, and often they far outweigh the homology-directed repair product, will be these indel byproducts. And each of these could have a different biological consequence. So that's really, I think, one of the most striking differences between prime editing and using a nuclease to introduce HDR. The DNA specificity of prime editing turns out to be unusually high for a CRISPR gene editing technology because instead of just having one kind of specificity check between the guide RNA and the target DNA sequence, there are actually 3 different specificity checks because of the complicated mechanism of prime editing. And as a result, if you look at the off-target editing, if you compare the off-target editing of Cas9 nuclease with that of prime editing, you can observe that the prime editors have far fewer off-target edits. The off-targets in this case are shown in yellow, orange, or red. Again, because we think that this mechanism provides 3 opportunities to check for a perfectly matched sequence before a prime edit can take place. So our lab and others have begun to use prime editing to install or correct pathogenic mutations that we could not before edit in human cells. For example, we can directly convert the normal hemoglobin sequence to the sickle cell anemia sequence, which requires an A to T change using prime editing, and then we can use prime editing to change it back to the normal sequence. We can precisely insert the 4 base pairs that are the most common cause of Tay-Sachs disease, and we can use prime editing to precisely remove these 4 extra bases. We can install a G to T mutation in the prion protein allele, which is proven to be protective for prion disease. And you can use prime editing to install a variety of small sequence tags, like epitope tags, or even recombinase, site-specific recombinase sites that then allow you to use a recombinase to insert a large gene into a site in the human genome that you choose. All right, then finally, I'll end by just summarizing some of the ways that we have more recently improved prime editing. So what I just described was basically the original system. And over the past couple years, we've identified the cellular bottlenecks that limit prime editing efficiency and engineered prime editing systems that make them more efficient. We began by improving these PEG RNAs. We realized that the PEG RNAs are vulnerable to being degraded. These 3' extensions are chosen by us, not by nature. So they are not evolved to resist degradation. And as a result, you can get these degraded 3' extensions, which are especially bad because not only can they not do prime editing, but they actually poison prime editing reactions by occupying the target site, even though they have no chance of actually resulting in a prime edit. So we showed that if you spike in these truncated RNAs, PEG RNAs, these truncated degraded PEG RNAs, that you poison prime editing. So James Nelson and Peyton Randolph, a former and current postdoc and graduate student respectively in the lab, discovered structured RNA motifs that protect that 3' end and improve PEG RNA stability. They turn out to be RNA pseudoknots, and these RNA pseudoknots greatly decrease the fraction of the PEG RNA in a cell that is degraded to the point where it can no longer mediate prime editing, represented by this light gray wedge. So in the engineered PEG RNAs, as we call them, or ePEG RNAs, where we've added one of these motifs to the 3' end of the PEG RNA, you can see that the PEG RNA preserves its structure, its extension, with a much higher frequency. And as a result, these ePEG RNAs improve prime editing efficiency in pretty much every cell type that we've tested thus far, including some primary patient-derived cells. And we now use these routinely as our main standard prime editing procedure. Next, we also went to improve the prime editing protein, or to understand what are all the ways that the cell responds to prime editing intermediates. And in collaboration with Britt Adamson and Jonathan Weissman's labs, Britt is an assistant professor at Princeton. This is work led by a graduate student in the lab, Peter Chen, together with Jeff Hussman, who was a postdoc in Jonathan Weissman's lab. We applied a really exciting CRISPR screening technology called RepairSeq, which allows you to knock down each of hundreds of DNA repair genes and measure the effect of each of those knockdowns on gene editing outcomes. And when you apply this screen to prime editors, you come to the conclusion that the best ways to improve prime editing in the screen are to decrease the cell's mismatch repair response. So why does this make sense? Well, in eukaryotic cells, mismatch repair, as I mentioned, is cued by the presence of nicks. We can't avoid nicks in prime editing because in order to do DNA polymerization, you need a nick. So you're always going to make this intermediate in which the edit in green is on a nicked piece of DNA. And if mismatch repair finds this intermediate, it will repair it into the original sequence because the nick is on the edited strand. So it makes sense that mismatch repair is bad for prime editing. So what we wondered is whether we could temporarily inhibit mismatch repair to give this intermediate a chance to get ligated into this complete heteroduplex. And then if mismatch repair sees this heteroduplex, at least with roughly equal probability, it should resolve into our intended prime editing outcome. So to make a long story short, we engineered a dominant negative MLH1 protein that can temporarily turn off or impede mismatch repair when we transiently express this MLH1-DN protein, as we call it. And doing so improves prime editing on average by more than 3-fold with the PE2 prime editing system. And so that led to our current state-of-the-art prime editors, which include PE4, which is basically the PE2 system plus the MLH1 dominant negative protein transiently co-expressed, and the PE5 system, which is the PE3 prime editor, which uses a simple guide RNA to nick the non-edited strand to help promote desired outcomes. If you combine that with transient co-expression of MLH1-DN, you end up with PE5. And these improvements can cause dramatic increases in the efficiency of prime editing in a variety of cell types, including, again, some patient-derived cells. Now, maybe my favorite part of this study was the realization that because we now know that mismatch repair impedes prime editing, we can simply design our prime edits in a counterintuitive way to make them natively resistant to mismatch repair. And the way this works, without having to even express MLH1-DM, the way this works is that mismatch repair looks for small mismatches, single base pair mismatches, maybe two base pair mismatches. But as you introduce additional changes, additional mismatches that you can install with the prime edit, this intermediate begins to not look like a substrate for mismatch repair because it has too much disagreement between the DNA strands. And so we showed that counterintuitively, if you install additional benign or silent mutations near the desired prime edit you wish to make, shown in green, you will actually improve the efficiency of prime editing. Normally, we think that making more mutations is more difficult, but here's a case where because we understand how the cell responds to prime editing intermediates, we can exploit that knowledge to improve prime editing without having to do anything other than introduce some additional mutations that are benign or silent into the pegRNA. And this really is representative of the way that prime editing has progressed. Like many gene editing technologies, there are certain sites and certain cell types in which prime editing is inefficient. And the original prime editing systems, PE2 and PE3, you can see in these three examples, which are all therapeutically relevant prime edits, we get very low efficiencies initially, sub 5%. But through systematic improvements using PE3, PE4, PE5, an optimized architecture called PE Max, the engineered pegRNAs I talked about, and now these benign silent mutations to evade mismatch repair, we can observe dramatic improvements in prime editing efficiency and, in many cases, decreases in the amount of indel byproducts as well. So I think this presents a very promising future for prime editing in which we now have an emerging understanding as well as improved components for prime editing that allow our lab and many others using prime editing to take on very challenging edits. And indeed, many papers have now been published using prime editing, even though we published it at the end of 2019, just before the pandemic sort of shut down a lot of labs, including our own. But Adgene has now distributed more than 5,000 prime editor constructs to more than 1,600 labs now. And there are more than 500 PubMed publications that use prime editing. All right, so to summarize this first talk, I introduced base editors as these engineered and evolved molecular machines that enable us to make these four kinds of single letter changes without cutting DNA. I showed how a different kind of base editor could be used to make the first purposeful changes in the sequence of mitochondrial DNA in living cells, and how prime editing is this remarkably versatile type of gene editing that, again, does not require a double-stranded break, but can directly copy small edited DNA sequences into a target site, and how it's advanced through a series of mechanistic insights and engineering efforts. All right, and that leaves me to thank a very talented group of students, postdocs, and collaborators. Andrew Anzalone, again here in the audience, led the development of prime editing. Some of these researchers I'll talk about in my next lecture. Peter Chen, in collaboration with Britt Adamson and Jonathan Weissman's lab, led the PE4, PE5 stories. Alexis Comore developed the very first cytosine base editor. Nicole Godelli developed the adenine base editor. Beverly Mock led the development of the mitochondrial base editor in collaboration with the labs of Vamsi Mutha and Joseph Mogus. And Peyton and James Nelson led the development of the engineered pegRNAs. And Tina Wan and Shannon Miller led the evolution of the Cas9 proteins that recognize new PAMs. And Michelle Richter and Kevin Zhao led the evolution of the highly active base editors. And thank you for your attention, and I will give a shorter and therapeutically oriented lecture in the second part of the seminar. Thank you.
Thank you very much, Professor Wu, for this very fascinating lecture and interesting presentation. I wonder if this, advances in fine editing, there is still a room for base editing technology.
Yeah, I think there will be. Base editors have just been their mechanism is very different, so they have different strengths and different weaknesses. And I think one of the strengths of base editors are that because they're directly making the change that you want by doing chemistry on the base itself, they are not as reliant on the cell to do these complicated DNA gymnastic steps. And as a result, base editors have worked more easily in a broader range of cells and are generally more efficient than prime editors, at least at the current generation stage. So these days, we expect that we can find base editors after some optimization that will edit at 80, 90+% now in cells in a dish, and often in animals to 30, 40, 50, 60, 70%, sometimes 80%, as I'll show you. So base editors currently are still, I think, more efficient and more robust in that they are more likely to work in more different cell types. But prime editors, of course, offer a versatility that if you want to make a T to D change or an A to T change or insert 2 bases or delete 5 bases, there really isn't a way that you can do that without relying on HDR, which again is very inefficient in most cell types other than prime editing. So I think they both have a role to play, and it explains why they're both heavily used, even though it's been 6 years since we published the first base editor, and now 2.5 years since we published the first prime editor.
Earlier today, I don't know if you can recognize me in the national press. First, I'd like to congratulate you again on winning the prize. And
thank you.
I have to say, I've been following your amazing work over the years, and it's one thing to read all this exciting science, and it's another to hear it from you directly in this elegant lecture. So thank you very much for sharing that with us. I was very fascinated by the idea of this accelerated Darwinian evolution, and I was just wondering if there is a way to learn from this to make it a little bit more supervised with machine learning in order to avoid the unpredictability with the randomness of evolution.
Right.
Evolution is the best engineer, right? But if you can tweak it even better with machine learning, I wonder what amazing things
right.
can happen for you.
Yeah, there are labs that are integrating machine learning into their protein evolution experiments. Our lab has done a little bit of that, but I think the best use of machine learning for true protein evolution now is to provide, is maybe twofold. One, to provide starting sequences that are most likely in the machine learning model's estimation to access parts of the fitness landscape that are highly active, that have highly desired properties. The other way that our lab has used machine learning interface with protein evolution is to take this massive amount of data that comes out of a PACE experiment, where we can get basically unlimited sequences. If you want to sequence 1 billion sequences that come out of PACE, you can do that because each of these lagoons have typically billions of different phage in them. And of course, they run continuously, so you can take time points as often as you want. So Max Shen, a former student in the lab, now at Genentech, he developed a machine learning model that can take this rich data set of high throughput sequencing data from protein evolution PACE experiments and quickly identify which clones are the most active. The most active clones are not necessarily the ones that are the most abundant. They are actually the ones that just have the highest fitness, which means given time and the static condition, they should outcompete the other genotypes. But at any one snapshot of a protein evolution experiment, which is often dictated by when the researcher needs to go home, when the researcher feels like enough is enough and wants to stop the experiment, it's not necessarily the case that the most abundant genotypes are the best ones. So his machine learning model called Evorical can do two things. First, it can reassemble from short read sequences entire full-length genotypes, because it's smart enough to say, okay, this sequence of 300 base pairs has mutations that covary exactly over time with this sequence, so they must belong to the same gene. And it can estimate a fitness for every genotype and tell you should focus your assay on these genotypes because they're the most likely to be fit. So that's been an exciting development. But I think in general, the intersection of machine learning and protein evolution is really a fertile one for future research, even though it's sort of young. It's in its infancy still.
Thank you, Dr. Professor. I have two questions. You might have heard about these. One is, my name is Dr. محاضرة and I'm Chief Vice Hospital. My first question is, what's long-term genotoxicity effect of these technologies? And my second question is, what's the best way to deliver these in vivo?
Yeah, sorry, what was the first? I missed the first question.
Long-term genotoxicity.
Oh, genotoxicity. Yeah, so genotoxicity, I think, can come from off-target editing, of course, and depending on the way that the genome is being manipulated, there's, of course, genotoxicity from certain kinds of manipulation, like making double-stranded breaks. It can cause chromosomal aneuploidy. So, with respect to the first question, obviously, we all want to deeply understand, assay as sensitively as possible, and engineer both editing agents and ways of delivering them that minimize off-target editing. It's probably the primary way to minimize the risk of genotoxicity. And also, I would say being very aware of what kind of editing mechanism you're doing and what are the ways that the cell might react that could be clinically relevant. Could be toxic. So FDA, of course, is very aware of this, and other regulatory agencies. And in some of the guidance they issued recently, they noted that if you're, for example, using a nuclease, you should look for chromosomal translocations because that's a known toxic consequence of a nuclease. And there may be similar consequences that are discovered for the non-cutting technologies like base editing and prime editing. But I suspect that many of the genotoxicities that researchers will worry about, at least in the short run, are those that come from cutting chromosomes, because that's a very rare event. There are, it's estimated that in every 24-hour period, each of your cells experiences more than 10,000 nicks. There's very, very common intermediates, but very few double-stranded breaks, fewer than 10. So the cell really responds differently to different kinds of surgery, if you will, on its genome. With respect to the second question, I'll answer that in my second talk, because I'll talk all about lots of different delivery methods for in vivo base editing and prime editing.
We have one more.
This is recently graduated from medical school. Actually, I have a question. Maybe Dima have to ask, which is regarding the relation between the water techniques and certain type of mutation. So, is there any relation between a certain type of mutation and technique should be used? Because maybe special nature of mutation has a certain type, it should be used by a certain type of technology. But this question came to my mind, so the relationship between the technology and the certain type of mutation.
I had a hard time hearing the question. I don't know if somebody closer could summarize it for me.
Actually, my question is related the certain type of mutation and technique should be used. Is there any relationship between the certain type of mutation, for example, for sickle cell anemia, thalassemia, cystic fibrosis, and the technique should be used?
Yeah, there's actually a reasonably simple emerging map. I'd say the map goes something like this. First, there are only 3 general ways to edit target sites of your choosing in mammalian cells. You can use a nuclease, you can use a base editor, you can use a prime editor. That's pretty much the case right now. If your desired change is a disruption of a gene, then you could consider a nuclease, especially if you don't mind the mixture of products that will result or the double-stranded break. You can also use a base editor or a prime editor to disrupt a gene, because you can install an early stop codon, you can get rid of a splice site, et cetera. You can get rid of a start codon. So there you have lots of options. If you wish to make a transition point mutation, so one of those 4 kinds of pyrimidine to pyrimidine or purine to purine changes, those are candidates for base editors. And in most cases, there's now a base editor that can make that edit paired with a proper Cas protein that can land on the site correctly. Sometimes you can't, because bystander editing, maybe there's 2 Cs right next to each other and you only want to edit one of them. That can be a challenge for base editors. And in that case, you may want to consider a prime editor. And then for everything else, I would say if it doesn't fit one of those first 2 categories, you should start with a prime editor.
One final question. I have a question about all of these primary things and base editing.
I saw that you did it in some of the primary cells, like the fibroblasts. My question about the genes, so like I'm interested in immunology, so I could see some of the genes are amenable to some of these base editing or prime editing tools. Do you think in the future we'll have like a specific gene or a certain gene that will be more efficient in this category compared to other? I'm thinking about the like doing it in vitro or in the clinical trial.
Yeah. If I understand the question, it's maybe about what is the relationship between the gene of interest and maybe the cell type of interest and the efficiency of editing. And it's very difficult to predict which genes are edited better or worse. As the editing tools have gotten better and better, as base editors became more efficient and now prime editors more efficient, we see less variation, because the sites that benefit the most from these improvements are the weakly edited sites. The ones that are already edited 60, 80%, if you use a more active editor, you generally don't get much improvement because they're already edited very efficiently. The ones that are edited, sub 5% benefit the most. So with base editing, we've seen a real compression of that range, where now, if you give me 100 sites and a few months to identify base editors that can edit those 100 sites, if I had a large enough team, I'd be confident I could probably edit 95 of those 100 sites, in cells in a dish with greater than, say, 50% efficiency. But I think that reaching that level of robustness is a fairly recent development, maybe the last couple of years. So I think prime editing is in the middle of following a similar course, where initially it was very efficient in certain cell types, like hex cells, and at certain very well edited sites. And now it's become much more robust at a variety of sites and in a variety of cell types, even though I think it's still, on average, less efficient than the most efficient base editors that we have.
Thank you very much, Professor Wu, for this lecture. And now we'll have a break for about 15 minutes. The other questions outside, and we will be back in 15 minutes. So, 8:45.
All right. Thank you very much.